So today we are going to discuss the post-transcriptional events in eukaryotes. We know that eukaryotes, after the transcription, there are certain processing. It's clear to make a final mRNA. So now, what are the processing? Before going to that, we must have a proper concept about the eukaryotic gene. Clear now? What is a gene? Gene is the basically the sequence of DNA that codes for RNA. So it is basically the sequence of DNA that codes for RNA. It may be tRNA, it may be rRNA, or it may be mRNA. Clear? So that is gene. Now, so you can see here, gene is the sequence of DNA that codes for either tRNA or rRNA or mRNA. Now, what is cistron? Cistron is that particular sequence of DNA which codes for a polypeptide. Clear? So, cistron is that particular segment of DNA that codes for a polypeptide. So, cistron is present within the genes which code for a polypeptide. Now, this cistron may be, so genes may be, according to the cistrons present, genes may be monocistronic and polycistronic. That means one gene may contain only a single coding sequence for a polypeptide. So that is called monocistronic gene. So within a gene we have a single coding sequence for a polypeptide, that's monocistronic gene. Or we may have polycistronic gene, where within the one gene we may have numerous coding sequence for various polypeptides, that is polycistronic, which we find in the prokaryotes. So monocistronic genes we find in the eukaryotes, and polycistronic genes we find in the prokaryotes. Clear? So now, monocistronic genes are the genes which contains the coding sequence for single polypeptide chain, and polycistronic genes are the genes which contains the coding sequence for numerous polypeptide chains. Clear? So where we find monocistronic gene? We find monocistronic gene in the eukaryotes, and we find polycistronic gene in the prokaryotes. Clear? So numerous polypeptide can be transcribed from a single gene in prokaryotes, but in eukaryotes generally a single polypeptide can be transcribed from a single gene. Next are the eukaryotic genes. So already we have learned that eukaryotic genes are monocistronic. Clear? So eukaryotic genes are monocistronic. Now this monocistronic, clear, eukaryotic gene may be split gene also. What is split gene? Split genes, so it contains a single cistron, but that is not, the entire sequence does not code for that particular polypeptide. Clear? There may be certain sequences within the DNA sequences which are coding for polypeptide. There might be certain sequences which does not code for polypeptides. So we can see here within the monocistronic gene we may have split gene, that means certain parts which are not coding for the polypeptide, those are called introns, and certain parts which are actually are the sequences which are coding for a polypeptide are called exons. Clear? So after processing, that is post-transcriptional processing, these exons appear in the mRNA, but these introns are removed. During post-transcriptional processing, these introns are removed from the RNA. Clear? So this is about the type of gene, and the gene why this is one of the reason why we require post-transcriptional processing, that is to remove the introns and to join the exons so that we can have a particular specific polypeptide. So now is the we will be discussing the post-transcriptional process. Now you can see here, this is the RNA which is formed after the transcription. Clear? This is the RNA which has been formed after the transcription, clear, which is called heterogeneous nuclear RNA or hnRNA. This heterogeneous nuclear RNA or hnRNA will undergo post-transcriptional processing for formation of messenger RNA or mRNA. So the gene, when it is been transcribed, clear, is not necessary that is directly forming mRNA in the eukaryotes. So it will first form heterogeneous nuclear RNA or hnRNA, which is consisting of introns as well as exons, clear, and this will be by post-transcriptional processes, it will be changed to mRNA, which will be functional for forming the polypeptide. So let us see how it occurs. So for our convenience of the study, we have divided post-transcriptional processes into three parts. One is the capping, clear, that's my addition of 7-methyl guanosine on the 5' end of the RNA. Tailing, that is addition of polyadenylate nucleotide sequences, clear, on the 3' end of the hnRNA, that's RNA. And finally is the splicing, that is removal of these introns. These are the introns, removal of these introns, and finally adding the exons, which to form a functional mRNA. So let us see how it occurs. So first of all is the capping. Where this capping is done? Capping is done on the 5' end. So this is the 5' end of the hnRNA. Clear? Now at first, this is the 5' end of the hnRNA. At first, guanosine triphosphate is added, clear, and guanylyl phosphatase breaks triphosphate, releases pyrophosphate, and guanosine monophosphate is added to the 5' end of the RNA. Clear? This is added to the 5' end of the RNA. So I am repeating once. So this is the 5' end of the RNA, which contains the triphosphate with a nitrogen bases, the nucleotides, clear. So triphosphatase breaks off one of the phosphate, clear, releases one of the phosphate from this triphosphatase, and now this is the condition. So now guanosine triphosphate is added, clear, by guanylyl triphosphatase, pyrophosphate is removed and guanosine monophosphate is added to it, clear, and with this guanosine monophosphate, now next part is the methylation. So 7-methyl, clear, methyl guanosine triphosphate is formed at the 5' end of the (7-methyl guanosine phosphate is added to the 5' end of the RNA, which is called capping, and this is required for the guiding this RNA to the cytosol. This is required for guiding this RNA to the cytosol. Next part is the tailing, that is the addition of polyadenylate chain on the 3' direction of the DNA. So we can see here this is the 3' end of the hnRNA, and here are some poly region, that is numerous adenylate region, and beyond that this is the non-coding region. So this portion is spliced off, and there is polymerase, clear, which add only adenylate, clear, the adenyl residue. So these adds going on adding additional residues of adenyl residues on the 3' end, and you can see a long chain of adenyl residues. Now why this happens? Actually this happens, clear, this actually happens to protect this RNA from the activity of nucleus, and we know that in eukaryotes it has to move outside the nucleus, it has to move to the cytosol, clear. So to protect this RNA from the activities of the various nucleus which may break this RNA, so there is a poly A tail, clear, to protect this RNA from the action of the nucleus. And this is now the last part, that is splicing. And that means, actually splicing means it is the removal of the introns or intervening sequences. Already we have done this. These are the intervening sequences which are non-coding sequences, clear. That means the protein will not be coded from these particular regions. So these removal of these intervening sequences, clear, and these are the exons which will be coding from the protein. So removal of these intervening sequences and addition of these exons is the splicing. Now actually splicing occurs by the splicing complex appears. The splicing complex are formed by the small nuclear RNA and protein. So small nuclear RNA and protein form the splice complex. In mammals we have spliceosomes. Now this splicing complex appears on both 5' and 3' end of the introns, clear. So suppose this is the intron, so in 5' as well as 3', in both ends there the splicing complex appears, and now this splicing complex, there is a turn and this splicing complex come together, clear, and this splicing complex ultimately come together, and finally it removes this introns from the DNA. So what happens, at first the introns are to be removed, splicing complex are formed by the small nuclear RNA and protein. These splicing complex are formed in both 3' end and the 5' end of the introns, and now these splicing complex which are there in the 5' end and the 3' end both comes together, clear, joins, and as a result they removes, clear, this, or separate these introns from the exon, clear. In mammals we find spliceosomes for this, and now finally these two exons will be attached, forming a long chain containing exons only. So thus now, so after capping, so 3 processes has been done. What is capping? That is addition of 5-methylguanosine on the 7-methylguanosine on the 5' end, then addition of poly A tail on the 3' end, and finally the removal of the introns from the hnRNA ultimately lead to the formation of a mRNA, clear. Now it is the mRNA with 7-methylguanosine triphosphate at the 5' end, polyadenylate tail at the 3' end, and these are the all exons attached together to form a long continuous exon, clear. So now this mRNA is to be transported to the cytosol, and which will be used for the formation of protein. So this is the post-transcriptional processes. This is how this post-transcription, after transcription, hnRNA, that is heterogeneous nuclear RNA, is converted to mRNA, which will be the functional RNA for the formation of the protein.